webinar. And that's great that you can attend. We also have the opportunity to schedule a one-on-one consultation with any of our instrument managers. I've highlighted in blue my contact information if you're interested or have a cryo-EM project you would like to discuss. And then lastly, we also have limited remote connection to data analysis and data analysis software if that's something that would help you with your research. Okay, so also similar to the previous webinars, just wanted to overview the instrumentation that's available at CEMAS. So obviously we have a lot in terms of electron microscopes, focused ion beam instruments, and also SEMs that are available. So I'm not going to talk in depth about all of this, but I'm mainly going to focus on our two cryo-EMs, which are the Glacios and the Titan Krios. So the main difference between those two microscopes would be that the Glacios is a 200 kV electron microscope, and the Titan is a 300 kV microscope that has a few more sort of extra, we call them bells and whistles, but sort of more advanced optics, like things like a phase plate, energy filter, but both of them are equipped with direct electron detectors, which I'll discuss in the slides later, but those are kind of necessary for doing structure determination, but both of our microscopes are equipped with those detectors. I also wanted to emphasize that nearly all of the high resolution structures that are being published today come from data that's collected on the Titan Krios. So we have one of the best microscopes available to collect your single particle data. Okay, so also at the beginning of these webinars, we sort of like to give some background information about who we are as presenters and instrument managers. And so just to give you a little bit of background about myself, I graduated from Ohio State with an undergraduate degree in chemistry. And then I moved on to grad school and did my master's in chemistry at Caltech, where I did research mostly sort of in the nanomaterials environment, and I did a lot of atomic force microscopy, as well as a lot of sort of surface chemistry. In addition, after that, I completed my PhD in biomolecular chemistry at Emory University in Atlanta. My thesis focused on biophysical mechanisms of notch activation, which is a pathway that's important in cell development. So there I kind of got more of a flavor of the biological side of things, and I continued to have an interest in systems that are mechanically activated, which is a little bit different than sort of your typical cell signaling. So when I moved back to Ohio State, I started as a postdoc researcher in the Sotomayor lab. That was for 4 years. And there I was trained as a structural biologist, mostly in X-ray crystallography and some EM. And then using those structures, I also conducted some molecular dynamic simulations and sort of expanded my interest in structural biology. So when the microscopes that I just talked about were built at CEMAS, I was really interested in getting involved in cryo-EM. And so I reached out to Dave and became a part of the CEMAS staff as a cryo-EM research associate. Okay. So now to get kind of more to the cryo-EM capabilities that we have at CEMAS. So I've kind of broken it down here into 4 different areas. The only area that I'm going to be talking about in depth today will be the single particle analysis, or the box in red in the upper left. There will be upcoming talks that will talk more about, for example, cryotomography. So Bin will be giving a talk later in May, where we'll talk about sort of tilting specimens and doing 3D reconstructions that way. Nicole will be giving a talk in 2 weeks about microcrystal electron diffraction, or micro-ED. This is a very sort of hot topic right now in cryo-EM, where very small crystals, nano or micro-sized crystals, can be used to collect sort of a processive electron diffraction pattern that can be used to solve the structure of small molecules or proteins. So that will be very an interesting technique that's sort of up and coming, I would say. The third box here that's sort of outlined and dashed is our cryo-fib milling capabilities. So this is something we're trying to currently build up. But for those of you that have very thick cells or tissues that you would like to look at using cryo-EM, your specimens need to be thin, so at least or close to 200 nanometers or thinner. And so that process usually would require fib milling, so using a focused ion beam to actually mill away portions of the cell until it's thin enough to actually be looked at in the electron microscope. So as I mentioned, I'm going to be talking mostly about single particle analysis. And when I say single particle, sometimes people get a little confused about what that means. So particle in this sense refers to a protein or a macromolecule. So it can contain DNA, RNA, different types of ligands or small molecules. And that particle has been cloned and overexpressed and purified from usually bacteria or mammalian cells. So it's a single species or sample that exists in solution. And if you look at this picture here on the left side, you can kind of see where they were back in 2013. If you look at the density, it sort of looks more like a blob. But as we're getting better and better at resolving this type of data, you can start to see secondary structure like alpha helices and also side chains in the density. So right now, sort of the record, you can definitely get sub 2 angstrom resolution for single particles, which is very impressive. Okay, so why is this method so important? So cryo-EM was definitely a game-changing tool for structural biologists for several reasons. First of all, you can look at your sample in its native state, which means there's no fixing, no staining required. You don't have to grow crystals like you do it for X-ray crystallography. And in addition, it requires a much less sample. So we're talking microgram quantities versus milligram quantities that you would need for X-ray or NMR. The samples themselves are essentially entombed in ice. So this is vitreous ice. It's not crystalline, it's amorphous. And in addition, cryo-EM is able to handle to some degree your sample heterogeneity, so different conformations and understanding the flexibility of your molecule. That being said, every tool has its limitations. So for cryo-EM, most of that is related to the size of the molecule. So if you look at this chart, you can kind of see once you go below 100 kilodaltons, the difficulty of determining the structure becomes much higher. Whereas if the sample is much larger, there's less issues solving the structure. There's also other limitations. For example, as I mentioned, all the specimens have to be thin, which means you have to create very thin layers of ice. And also, because it's a biological sample, you have to be very careful about how many electrons you expose your sample to, because you will cause radiation damage at some point. So you have to keep the electron dose very low. There's also different issues I'll talk about related to sample preparation that are critical and can be bottlenecks to solving your structure. Okay, so just to go over the Cryo-EM workflow, for those of you that are interested in getting started in Cryo-EM but haven't started exploring yet experimentally, I've highlighted in red here the steps that I would say require the most time and effort. And so the first step is obviously the biochemistry aspect. So as a user, you would be responsible for expressing and purifying the protein or particle that you're interested in studying. And so that can often take many months or years to do. So once you have your purified sample in hand, the first step or first part of the workflow is to do negative stain, which is actually fairly quick. So only would take a few days to actually do. But it's very critical in terms of your initial screening. In this case, the sample mixed, it's stained with a heavy metal, usually uranium, and then we image it. Typically in the case of CEMAS, we would use the Tech-9 30 to take images of your sample. So what you're looking for is a homogeneous sample with no aggregation. You want to see, sometimes it's nice to see different views of your protein. You could also, if you have enough of this, make a 2D model and try and do some initial classification if you have enough data. But it's a very important first step is to do negative staining. And I also want to emphasize that all of this workflow can be done at CEMAS. So we have the capability to help you through all of these steps, but that is definitely the place to begin is negative staining. So once that looks acceptable, then you can start thinking about how you're going to prep your sample for cryo-EM. And so here you can see the instrumentation that's associated with each of these steps. I'll talk about the Vitrobot in a couple more slides, but essentially the Vitrobot will help you vitrify your sample, so freeze your sample, and then we would screen on the Glacios and optimize. So we'd go back and forth a few times to do that. And once you have a good sample, we could collect data on the Krios, and then you can move on to the reconstruction of your particle using sort of different data analysis tools. Okay, the other thing I wanted to mention real quick is that the Glacios and the Krios are both equipped with an autoloader system. So you can load up to 12 grids or samples per session. And it's important to remember that the main thing is once the sample is frozen, it needs to be kept at cryogenic temperatures all the way through data collection. Okay, so how do you actually prepare a sample? So the most common substrate or grid that is used for cryo-EM is what's called a Quantifoil. So most of you hopefully have some familiarity with electron microscopy. So you have seen a TEM grid, right? So the standard size is 3 millimeters in diameter. The actual mesh size is usually between 200 to 400. Just depends on what type of samples you're looking at. And then the grid material itself tend to be copper or gold. Those are the most common for cryo-EM. So you have a 3 millimeter grid here. On top of the grid is what's called a foil material, which in most cases is amorphous carbon. So that's this gray sort of network with holes in it, you can see. So these holes, if you look in panel C here, are of a very consistent size. So this one is 1 micron in diameter. And you can see that each square contains hundreds of holes inside. So the idea is the water layer with your protein will span that hole and essentially create a very thin layer of particles that are frozen and can be imaged by the microscope. Okay, so back to the Vitrobot here. So we're using the Vitrobot as an example because that's the type of plunging system that we have available at CEMAS. There are other options. For example, there are manual plungers. Also, Leica and Gatan have similar products. I'll talk a little bit about Spotaton and Chameleon when I'm done because it's a little bit different method. But all of these first three here rely on blotting your sample. So you take one of the grids that I just showed from the previous slide, you place it on the tweezers that kind of come down the center of the Vitrobot, and then you apply 3 to 4 microliters of your sample onto the grid. And so here you can see the holes in the carbon. And filter paper will come on both sides in the case of the Vitrobot and wick away the excess liquid. And then very quickly, the tweezers will be plunged into liquid ethane. So that's what's going to freeze your sample very quickly. And again, this is sort of the side view of what you hope to see. So the carbon support, and then a hopefully less than 200 nanometer, so thick layer of ice with your particles will be formed there. So as I mentioned, the Chameleon is a little bit different. That actually uses like a spray technique, no blotting, to place the particles in the holes. So the overall goal of this whole sample prep instrumentation is just to form a nice thin layer, right, with intact particles. So you don't want denatured particles, you want intact particles that are inside the center of the hole. So that's the whole point of this process. And then, as I said before, I wanted to emphasize that once the sample is frozen, it needs to stay frozen or at cryogenic temperatures all the way through to data collection. So from this point forward, everything will be kept in liquid nitrogen. The other thing I wanted to point out is that this whole process of vitrification, this technology, was developed by Jacques Dubochet, who, along with Richard Henderson and Joachim Frank, won the Nobel Prize in 2017 for cryo-EM. So this part of the process is actually very important to getting cryo-EM to work. Okay, so once you have your samples prepared, what do they look like, right? So a couple things when people who look at, have never seen images that come from a cryo-EM notice when they look at samples. Initially, they're probably a little bit disappointed because the contrast doesn't look great. But if you think about it, your sample is essentially made of very light atoms, right? Carbon, oxygen, nitrogen. So it's hard to get contrast because the contrast is all basically based upon phase and not amplitude. So you'll notice, as I mentioned before, the larger the molecule that you're working with, or the particle, the easier it is to see in the ice. Okay, so this is just a series of images that were all taken at the same magnification, different molecular weights. So in the top left, you have 1.6 megadaltons. As you go across, you can see the particles are getting smaller. Hopefully you can also see different orientations of the particles. That's important. You want to see nice even spacing. So you want to see them spread apart, not too close to one another. A funny thing is going on here in this bottom left panel of the catalase. So you'll notice actually here in the center, the ice is too thin. So the ice is so thin that the particle cannot enter into that area. And that's an interesting effect that you do tend to see sometimes. You would anticipate that the ice is actually thicker towards the edge of the holes and then thinner in the center. So as the particles get smaller, you'll notice once you get down to something the size of hemoglobin, which is about 64 kilodaltons, the particles are just basically tiny dots that are hard to pick out. And so that's kind of one of the reasons why it gets more difficult as your particles get smaller and smaller. Okay, so these are obviously sort of perfect images of samples. So what in reality do they actually look like, right? So usually you'll see a variety of things. In panel A here, what you see are two different particles. One's 180 kilodaltons and the other one is 110 kilodaltons. So here you can see in the insets are just sort of blown up regions, but you can see individual particles. So the ice thickness here is good. And I can still see some contamination or aggregation, maybe some ice in there. But overall pretty good. So you could move on with a sample like sample A. In panel B, as I mentioned before, the ice is getting quite thin in the center. So what you'll notice is some areas are denatured. So the top and bottom portions, you'll notice you see something fuzzy. That's probably your protein that has just piled up there. And then in the center, you don't really see anything. And it's most likely either denatured or just empty. In panel C, again, this one was where the ice is too thick. Clearly you can see something is there. It doesn't look like a particle, it just looks like a tangled sort of mess. And this is not what you want to see either. You cannot move on with this sample. And then lastly, panel D. Panel D, you can kind of see what vitreous ice looks like. It has sort of no features to it. On occasion, you may see what you think is a particle, but it's unclear. It's obviously that there's not enough to look at in that sample. You can also see other things. So one of my users, I'm just going to use him as an example, but Kai from Mark Foster's group also has a different type of problem, which is a severe preferred orientation issue. So his samples don't like to be in different views, and they can actually order themselves almost in a 2D crystal, like in the corner here. So there are sort of a multitude of problems and issues that can arise from sample preparation. So I pulled this quote from the reference here at the bottom, which is a very recent review article that that just came out, and it says that, you know, still the limiting factor to achieving high resolution reconstructions is sample preparation. So getting a sample that is good enough to collect data. Okay, so a lot of this part, so sample preparation and data processing, those two parts of the process alone could probably be their own webinars. So unfortunately, I'm not going to have a lot of time to talk about them in detail. But other than to say that that these two processes are very intense and involve a lot of man or person power, I would say. So a lot of your graduate students' time would be devoted to these two areas, so data processing and sample preparation. So let's say you had a sample like in panel A in the previous slide that looked good and was suitable for data collection. So what would happen? You would put your sample into the Krios and set up a data collection. So over 1 to 3 days, you can collect, let's say, probably roughly 3,000 movies or raw movie files per day. So that kind of gives you an idea of the total amount of data you're collecting. So that would just be these compressed raw movie files. Okay, so after that, what happens next is what is called sort of like a pre-processing step. So this portion is not terribly time consuming, but it does require a lot of computational resources. So each of these movies, so what you get with cryo-EM data is not just a single snapshot. You get a movie of your particles. And what happens is these movies are actually kind of blurry initially. So you have to do a step called motion correction, where you align each individual frame of the movie to produce a nice clear image of your particles. Okay, so that's step one, is motion correction. After you do motion correction, then you can fit the CTF or contrast transfer function, which I'm not going to talk about here, but essentially it's going to tell you how high of a resolution the information in your images is. So essentially, what is the best achievable resolution you could get for the data that you've collected? So you want to fit the CTF to your images and find out how good is this data. After you do the CTF fit, then you can go back to your images and go and actually manually pick particles. So each of these yellow circles here represents a single particle, right? And they don't have different views here, but sometimes they're in different orientations. So it's actually you're going to pick some manually. Generally speaking, you train the particle picking algorithm, and then it will go through automatically and pick the particles for you. Okay, so then it picks all of these particles and extracts them out of your images. And then the next step after that is it's going to take all of the particles that you've picked and try to sort them out. And that is a process called 2D classification. Okay, so this could involve hundreds of thousands to a million particles that it's going to go through and classify. And then after 2D classification, you can start to build an actual 3D model of your particle that you're interested in. So like, as I mentioned, most of the high resolution structures, I would say, would be contained between 50,000 to 200,000 particles. So you're not choosing all the particles that you pick here, but you're going to do some curation during this process until you can get a good enough set to build a model. Okay, and I also wanted to emphasize here that the new algorithms and reconstruction strategies are actually part of the reason why the other two Nobel Prize winners were awarded a Nobel Prize in 2017. So I also wanted to say at CEMAS, we can do similar types of things. I'm not going to talk, this is just data we've collected from avoferritin, which is a standard sample, but we have been able to reach sub-3 angstrom maps using our instrumentation. Okay, so kind of want to make sure I leave enough time to talk about the interesting cryo-EM results that have come out about the SARS-CoV-2. So I just wanted to emphasize again, so two of the reasons why this resolution revolution happened were, one, data processing improvements, right? So all the software and algorithms that have been improving over time. And then also secondarily, our hardware improvements. So our Krios is equipped with a K3 detector, which is a direct electron detector. So both have a direct electron detector, but essentially there are sort of no, once the electron hits the CMOS film, then you will read out your image. Whereas in a conventional CCD detector, you will get essentially some, I'm trying to think of what the best word is, some, not blurring of your image, but the image will not be as clear as it is with a direct electron detector. So this advance in hardware is another reason why. In addition, the K3 has a much larger field of view, so you can get more particles in each picture, and it's much faster, which means you can just collect a lot more data. Okay, so that's kind of the main portion of sort of the cryo-EM, or the bulk about the process and the workflow. So how does this help in current research? And so there's a very obviously good example of how cryo-EM can advance biomedical research. Right now that's going on with SARS-CoV-2. So hopefully I'm not going to information overload you. I'm not a virologist or an immunologist, but I just kind of want to give you this short, interesting story about the results that have come out very recently. So most of you know about the structure of this RNA virus, so SARS-CoV-2. So it's an RNA virus, right? So inside, encapsulated in here is the RNA. It's decorated with these spike proteins, which you see in red. So those are actually how the virus is going to enter into cells, right? So if you look in the right-hand panel, the spike protein binds to what is called the ACE2 receptor on the host cell. And then once it gains entry into the cell, it hijacks the cell machinery to essentially produce more copies of the virus, which are then released back into other host cells. So this all happened very quickly. In January 2020, they released the complete sequence of this virus, which is about 30,000 nucleotides, into GenBank. And very quickly, structural biologists started targeting the spike protein, right? Because they know that that's going to be a key target for vaccines and therapeutic antibodies and diagnostics. So I will say that the researchers who had already been involved in studying coronaviruses previously had a very distinct advantage in determining the structure of this spike protein. So they had a lot of the methodology down already. And so a couple key things I want to say about the structure. So I know many of you are probably not structural biologists, but the spike protein itself is a homotrimer. So each monomer is about 200 kilodaltons. It's also glycoprotein, which means it has sugars that essentially decorate the protein. So there's 22 sugars per monomer. That'll become important, you'll see in the structures that I show you. It also has this furin cleavage site, which does not exist in SARS-CoV, or the original SARS-CoV. So it may have a slightly different mechanism of activation. And it also does bind with greater affinity to the receptor. So that could be why it may be more infectious than SARS-CoV. Okay, so this is very impressive, I think. So the McClellan lab at UT Austin were the first to clone and express and purify the spike protein. So as I said, in January, the sequence was released. They submitted this manuscript to Science on February 10th, 2020. So that was maybe a little over a month after they received the sequence. After purification, they did 24-hour data collection on the Krios with the K3. So they got about 3,000 movies out of that data collection. And from that, they could determine a 3.5 angstrom resolution structure of the spike protein pre-fusion. Okay, so that looks like, so again, when you look at this cryo-EM image, it's not terribly impressive, but if you think they had 3,000 movies, from those movies, they picked about 600,000 particles. Here you can see the 2D classifications, right? And so from there, they curated it further down to about 200,000, 250,000 particles. And from those, they were actually able to build their ab initio models. There is heterogeneous refinement because there's multiple conformations of this structure. So if we go to this, we can actually see, so this is the map that came from that data. So in white and gray, those are the other monomers. So you see in ribbon, there is one monomer that's sort of outlined there. The green domain is the receptor binding domain. And in this case, this is in the up conformation. So this portion is sticking up. It can also be in a down conformation. So they were not the only ones that were interested in the spike protein. There were two other groups that were very, very close in solving the structure. So the Visser lab from the University of Washington also solved the spike structure. This was submitted to Cell in February, also February 19th. And then another group from Westlake University in China actually solved the structure of the spike bound to a portion of the ACE2 receptor. So that was submitted in February 12th. Okay, so there's a really cool video here that was put together by someone at the Cryo-EM Slack facility. So I'm just gonna play this. These are structures from the Wiesler paper. They released their coordinates to a public database. So they were actually able to kind of put this animation together. So on the left is the closed. So I mentioned there's two conformations. There's a closed state and an open state. So left is closed, right is open. So I'm gonna start this movie. So you see it's a trimer. Down here is where it binds to the membrane. Up here is where it's gonna bind to the ACE2 receptor. Okay, so here in different colors, you can see each monomer and sort of how it wraps around its neighbor. Okay, so they're gonna superimpose the up and the down conformation. And then there'll be a nice little animation here where you see it's closed and then swings open, closed, and then it's gonna turn. Okay, so in the up or open conformation, what you'll see here is that there's a crystal structure of part of the ACE2 receptor that binds to the spike, and they're gonna dock it to the spike. Let's see if I can get this to go. Okay, so it docks to the spike, and this is the interaction that you would want an antibody or something to block, right? So you don't want this to happen, otherwise the virus can enter into the cell. Okay, and so here you can kind of clearly see where it's actually binding. Okay, so I thought that was a pretty cool animation that they put together. Another thing that I think is interesting to keep in mind is that the structures that I have shown you previously, they're basically like snapshots, right? They're photographs of the spike protein, but in reality, the spike protein is very dynamic, and so there is a research group called D.E. Shaw Research that uses computational resources to simulate what the spike protein or how it behaves essentially in a physiological state. So it's solvated, there's ions present, and you'll notice here there's a lot of little orange dots. I'm going to start the video. This is the open state, right? So you're going to see these little orange circles here, those are all the sugars. So I mentioned it's highly glycosylated, and you can kind of see how the sugars stick out. This is very common for viruses to have this glycoprotein type of shell outside. But the thing that you'll notice is that it's very dynamic. It swings open and closed. And you'll also notice how are you going to actually design something to either come in and block. So you have to consider all of these motions that are happening and how that would be difficult to rationally design. But this gives you a good model for what is actually happening inside of the cell. And they have made these all publicly available. So if you're interested in molecular dynamic simulations, you can actually take a look at these structures as well. Okay, so that is my presentation. I'm sorry I went over time a little bit, but I just wanted to once again, sorry, let me go back and plug our upcoming events. So we have a continuation of this discussion series. Next week, as I mentioned, will be Steve Buna, who's one of our SEM managers, talking about SEM and EDS mysteries. And then the following week will be Nicole talking about micro-ED. So you can always find more information about the events that we're hosting at this website, and other ways to reach out to us. All of these websites, I put my Twitter handle here as well as the one for general CEMAS information. We're on Facebook, LinkedIn. So definitely reach out to us if you have questions or project ideas. Now is a great time to reach out to us and see what we can do together.